All the topics we discussed so far this semester are the background for the main topic of this course, specifically biocompatibility. The topic of Lecture 24 is Evaluation of Biocompatibility, a short introduction. The study of biocompatibility must start with a reminder regarding the ultimate clinical objective for implants. Specifically, in order to be successful, implant materials and devices must succeed in forming new functional tissue, both in vitro and in vivo, without any adverse effects. To induce successful tissue wound healing processes is another requirement, and this of course follows the implantation of prosthetic devices, and the ultimate desirable objective in this respect is normal tissue regeneration and formation of functional tissue. Biomaterials biomedical engineers need to address these requirements and objectives from the material perspective. Another reminder of an earlier definition of biomaterials, which are substances other than food and drugs, which are contained in therapeutic and/or diagnostic systems. How was this particular definition of biomaterials applied in the past? Biomaterials were developed using trial and error approaches. We reviewed some examples of polymeric elastic materials which were used for prosthetic devices in the vasculature, and metals which were used in applications in bone. Those were orthopedic dental implants. All of these approaches use materials that existed or they had been developed for other purposes, which were not specific to biomedical needs and specifications. It is not a surprise, therefore, that in most cases such applications were not successful. Failures of the trial and error approaches in developing biomaterials gave rise to several questions. One of them was, how does a material become a biomaterial? Is it possible to, for lack of any better word, transform traditional materials into biomaterials? How can this outcome be achieved? Attempts in the past regarding biocompatibility of implant materials achieved the following: to produce biomaterials which were non-toxic, non-pyrogenic, non-allergenic, non-thrombogenic, and you may add a few more specific characteristics in the non-something category. Therefore, these materials did not kill cells and tissues, did not induce fever, did not cause allergies, did not bring about excessive and uncontrollable blood clotting. Most recently, to this long list of non characteristics were added other specifications. Non-mutagenic, that means to change the type of cells or tissues. Non-oncogenic, not to cause cancer. Non-teratogenic, to create all kinds of other creatures as a result of the presence of the material. Not to elicit foreign body reactions, etc., etc. You can see that this list now becomes not only longer, but it addresses issues which are pertinent to cell biology, to cell biochemistry, all the way down to gene level issues of biology and physiology. Therefore, now, in the present, a biocompatible material is one which promotes biological processes that bring about, achieve the intended aim of the prosthesis. In other words, the material exhibits biofunctionality specific to the anatomical location and organ where the prosthesis is implanted. Bringing all these concepts from the past and present and projecting into the future, biomaterials now need to be, some people have called them quote-unquote proactive, because they are required to elicit specific, precise, select, rapid, and timely responses from surrounding proteins, cells, and tissues. Such events are known and referred to as interfacial interactions. Going back to the questions which started the present discussion, the objective of finding out how a material becomes a biomaterial now takes a very specific direction, namely, how can we achieve this particular outcome? That implies that processes need to be developed and used in order to determine the biocompatibility of materials. A major requirement in establishing the biocompatibility of materials is stated in this particular slide. Just because we designed and formulated and synthesized a successful biomaterial does not mean that the process is complete. This process needs to have, as the next step, evaluation of the biocompatibility. Most important, this particular process has to result in evidence of successful performance of our biomaterials, specifically under conditions that simulate those which will be the end-use application environment and time requirements. Moreover, the biocompatibility evaluation has certain requirements because there are crucial consequences if those requirements are not met successfully. Specifically, a biomaterial and the implant devices which are composed of those biomaterials must always be safe for the patients. They must be safe to the healthcare providers, and they must comply with regulations of various regulatory agencies, such as, for example, the Food and Drug Administration, also known as FDA in the United States. Determination of the biocompatibility of implantable materials and devices is an unavoidable requirement. It involves a series of tests which have been developed with the help of biomaterial scientists and engineers, and by taking into consideration the requirements by the various regulatory agencies. The process is an integral part of developing materials and devices for implantation, which will reach the market and will be used eventually by the healthcare colleagues to help patients. The processes involved are complex and are both in vitro and in vivo tests. In the case of the in vivo test, both local and systemic responses in the body are monitored. The biocompatibility determination, of course, helps us understand the biological responses involved, but all of these tests cannot ensure that this material and device of ours will perform optimally in human subjects. This is a major limitation of this particular process, but the process of determining the biocompatibility of implantable materials and devices is still appropriate and needed. It is needed, of course, because it is required by national and international regulatory agencies. I mentioned the Food and Drug Administration or FDA in the United States of America, but these days various countries have their own regulatory agencies, and if a product of ours, of the company in which we work, is aimed at being sold, let's say in Europe, the appropriate regulatory agency of the European Union needs to be consulted. Or if the product is meant to be used in other countries like Japan or Korea, etc., we must know that these countries have their own regulatory agencies, and therefore our implantable materials and devices need to meet the specifications and the requirements of the respective regulatory agencies. Who conducts the biocompatibility studies or tests? These tests are conducted in laboratories, in universities, in the research and development divisions of various companies in industry. In the past, most companies which developed materials and devices had their own research and development division. More recently, because of major changes in how various companies conduct business, many of these companies do not have research and development divisions anymore, but the big either implantable devices companies or pharmaceutical companies, they do still have major research and development divisions in their companies. Last but not least, these days, there are commercial companies which do that for their business. Therefore, if you and I have developed a particular material and device, but we don't have this capability either in our laboratories at universities or in the company where we work to do all these biocompatibility tests, we may hire these particular companies and they can conduct any of the tests needed, either in vitro or in vivo, I mean by that animal level experiments, for, of course, for an appropriate payment. They can do a very good job, and they are always present at various professional conferences. They have booths, and if you are interested, you can always go by and discuss the services that these companies provide, pick up copies of their brochures, ask them about what fees they charge, and decide whether you would like to have a specific company run the biocompatibility experiments for your implantable materials and devices. It is important to keep in mind certain prerequisite tests in evaluating biocompatibility. Specifically, characterization of the materials and devices in vitro under conditions which test the functional performance of the prototypes is a prerequisite. That means those tests have to be performed and completed successfully before any evaluation of their biocompatibility is even considered, never mind conducted. Keep in mind that materials and devices which do not pass the prerequisite test should not be evaluated further for their biocompatibility. Even if we are very attached to our favorite material and device which we designed and formulated and try to test, even when we are so attached to a particular material and device, we have to keep in mind the fact that the biocompatibility tests are extensive, they are expensive, and they have to be conducted only when all the prerequisite tests, which we're going to review in a following lecture, all the prerequisite tests have been completed successfully. Otherwise, there will not be any biocompatibility tests conducted. The evaluation process of the biocompatibility of materials is classified under the following three categories. First and foremost, a series of in vitro tests, followed by in vivo animal tests, and last but not least, clinical trials. The successful completion of the in vitro test becomes the rationale for following and doing the in vivo animal test. The completion successfully so of the in vivo animal test becomes the rationale and the reason why we were going to consider clinical trials after, of course, appropriate approvals from the Food and Drug Administration and similar regulatory agencies in other countries are consulted and given permission to do so. The ultimate objective in the evaluation of the biocompatibility of materials, of course, are the clinical trials, because this is the time when our favorite materials and our favorite devices are tested in the environment for which they were designed, namely inside the human body. This is the end of lecture 24, Evaluation of Biocompatibility, a short introduction.